Determination of primary productivity in pond water using light and dark bottles. A. To estimate the primary productivity in pond water using dark and light bottles. Primary productivity of an ecosystem is defined as the rate at which the radiant energy is stored by photosynthesis of a producer organism into the organic substances which are used as food materials. Gross primary productivity or GPP. This is the total rate of photosynthesis including organic material lost by respiration during the period. This is the total rate of photosynthesis including organic material lost by respiratory activity during the period. GPP is equal to total organic matter plus respiratory loss. The net primary productivity, this is the rate of storage of organic matter in the plant in excess of that lost by respiration during the period. NPP is equal to new organic matter by respiratory loss. Plants produce new carbon organic by the process of photosynthesis. In this process, oxygen is released by plants. So, by estimating the oxygen which is released by the plants in photosynthesis, the primary productivity can be estimated. In this method, light and dark bottles are used for estimation of primary productivity. Initially, the dissolved oxygen is determined. Then, after a finite time, the dissolved oxygen of the light bottle and dark bottle is determined. In the light bottle, the level of dissolved oxygen increases than initial level. In the light bottle, the level of dissolved oxygen increases than initial level. Because of this bottle, photosynthesis occurs. The increasing volume of dissolved oxygen in the light bottle can be determined by subtracting initial value of dissolved oxygen. The increasing volume of dissolved oxygen in the light bottle can be determined by subtracting the initial value of dissolved oxygen. This value is multiplied by conversion factor for carbon, which is 0.375, so that gives net primary productivity or NPP, because the value of respiratory loss R can be determined by estimating the dissolved oxygen D. In dark bottle, value of photosynthesis does not occur, so oxygen is not released, instead respiration occurs here. The subtracted from the initial value of dissolved oxygen, it gives the value of respiratory quotient R. If the value of R is added to the value of net primary productivity, it gives the value of gross primary productivity, GPP. Materials required, we need light bottles, dark bottles, rope or other means to suspend pairs of bottles in the pond, pipette, burette, reagents, conical flask, measuring jar, dropper. Reagents, manganese sulfate solution or MnSO4, alkaline iodide solution or Ki, starch solution, concentrated sulfuric acid or H2SO4, sodium thiosulfate, stock solution Na2S2O3. So procedure, fixation of dissolved oxygen. Fixation of dissolved oxygen must be performed in the field immediately after the sample has been secured. The dissolved oxygen is mixed by the following procedure. Take the water sample in 250 ml volume sample bottle. Remove the stopper from the sample bottle and add 1 ml of manganese sulfate solution by inserting the tip of pipette just below under water surface. So fixation of dissolved oxygen must be performed in the field immediately. So take the water sample in 250 ml volume sample bottle. Remove the stopper from the sample bottle and add 1 ml of manganese sulfate solution by inserting the tip of the pipette just below water surface. Okay. Remove the stopper from the sample bottle and add 1 ml of manganese sulfate solution by inserting the tip of pipette just below water surface. Then add 1 ml of alkaline iodide solution in the same manner as for manganese sulfate. 1 ml of alkaline iodide is added similarly. Then here you could see the result, which we will be learning after some time. Replace the stopper and mix the sample by inverting the sample bottle several times. Allow the precipitate to settle for a few minutes. After setting off the precipitate, cleaner fluid should occupy the upper portion of the liquid bottle. Using volumetric pipette, add 1 ml of concentrated H2SO4 by permitting it to run down the neck of the bottle and mix well by inverting bottle several times, at least 40 times. Allow the sample to stand for at least 5 minutes. Then titration, take 50 ml of sample in which dissolved oxygen has been fixed in a volumetric flask and titrate with 0.01 normal sodium thiosulfate solution to a very pale yellow color. Now add 1 ml starch solution and note the color of solution turn blue. Continue the titration until the blue color disappears. Record the used volume of sodium thiosulfate in 1 ml. 1 ml starch solution and note the color of solution turn blue. Repeat for concordant value in both light and dark bottle. Then determination of net primary productivity. Allow the precipitate for settle for a minute. Then after setting the precipitate, clear fluid should occupy the upper portion. Then the added concentrated H2SO4. Then take 50 ml of sample in which dissolved oxygen has been fixed in a volumetric flask and titrate with 0.01 normal sodium thiosulfate solution to a very pale yellow color. Now add 1 ml starch solution and note the color of solution turn blue. Then continue the titration until the blue color disappears. Record the used volume of sodium thiosulfate in 1 ml. Repeat for concordant value in both light and dark bottle. Determination of net primary productivity NP L minus I gram per liter 3 hours. L value of dissolved oxygen in the light bottle. I initial value of dissolved oxygen. Then determination of respiration R. R is equal to 1 minus D G per liter. I minus D. I minus D initial value of dissolved oxygen minus volume of dissolved oxygen in dark bottle. Determination of respiration is equal to R is equal to I minus D. I am initial value of dissolved oxygen, volume of dissolved oxygen in dark bottle. Determination of gross photosynthesis. Gp is equal to Np plus R mg per liter per 3 hours. Np is equal to value of net photosynthesis. R is equal to respiration. Once again, determination of gross photosynthesis is equal to Gp is equal to Np plus R mg per liter 3 hours. Then Np volume value of net photosynthesis, R respiration. Then determination of primary productivity. Primary productivity can be determined by multiplying the rate of photosynthesis by conversion factor of carbon, which is 0.375. Once again, earlier we mentioned about determining the respiration. Determination of net primary productivity. Determination of net primary productivity. Np is equal to L minus Ig per liter per 3 hours. L is equal to value of dissolved oxygen in the light bottle. Value of dissolved oxygen Np. Determination of net primary productivity. Value of dissolved oxygen in the light bottle. Initial value of dissolved oxygen. Then determination of respiration R. R is equal to 1 I minus D gram per liter 3 hours. I is equal to initial value of dissolved oxygen. D is equal to value of dissolved oxygen in dark bottle. So we got NPP. Yes, NPP we got. After determination of gross photosynthesis, Gp is equal to Np plus R mg per liter per 3 hours. Np is equal to value of net photosynthesis. R is equal to respiration. Then determination of primary productivity. Primary productivity can be determined by multiplying the rate of photosynthesis by conversion factor for carbon. NPP is equal to Np into 0.375 gram per meters cube per hour. GAP is equal to Gp into 0.375 G per m cube per hour. Result: gross photosynthesis 4 gram per liter per 3 hour. Net photosynthesis 2.8 mg per liter per 3 hour. Respiration 1.2 mg per liter per 3 hour. Net primary productivity or NPP 1.05 gram per m cube per 3 hour. Then gross primary productivity 1.5 gram per m cube per 3 hour. Okay. Then some earlier aspects related to the class calculations is also there. So we need to learn that. Some other aspects like volume of sample ml, initial sample, light sample, dark sample, burette reading, same initial final portions are there. Then volume of Na2S2O3. Like this. Initial sample, volume of water sample 20, volume of Na2S2O3 E2 0.4 ml. Normality of Na2S2O3 0.01 normal. Normality of water sample N1 is equal to N2 V2 by N1. (0.01 into 0.4 by 20. That is the normality of water sample. An equivalent weight of oxygen 8 gram. Amount of dissolved oxygen N into equivalent weight into 1000. N2 into E2 by N1. Normality of Na2S2O3. N2 into E2 by V1. Normality of water used is equal to normality of Na2S2O3 into volume of Na2S2O3 by volume of water. Normality of water is equal to volume of Na2S2O3. Volume of normality of water sample is equal to normality of water sample N1. Normality of water sample N1 is equal to normality of water sample N1 is equal to normality of water sample is equal to normality of Na2S2O3 into volume of Na2S2O3 by volume of water used. We get the normality of water. Okay, equivalent weight of oxygen 80 gram. So amount of dissolved oxygen N into equivalent weight into 1000. Don't forget that 1000. N into equivalent weight into 1000. You got that. Mg per ml. Mg per liter. Okay. Because we are calculating this in liter. Then light bottle, volume of water sample 20 ml V1. Volume of Na2S2O3 used 1.1. That is V2. Then normality of Na2S2O3 N2 is equal to 0.01 normal. Normality of water sample N1 is equal to 0.01 into 1.1 by 20. We got the results 0.00055 normal. That means normality of water sample is equal to normality of Na2S2O3 into volume of Na2S2O3 used by 20. 20 is the volume of water sample. Equivalent weight of O2 8 gram. Same is the case with carbon dioxide bottle. Amount of dissolved oxygen in light bottle is equal to N into 8 into 1000. Okay, mg per liter. Dark bottle, volume of water sample used. Volume of water sample used. Volume of water sample used V1. Volume of Na2H2O3 used. Normality of Na2H2O3. Normality of water sample. Normality of water sample is equal to volume of Na2H2O3 used into normality of Na2H2O3 by volume of water used. Same is the case with dissolved oxygen and dark bottle into 8 into 1000, 0.4 mg per liter. Then determination of net primary photosynthesis, which is NP. Determination of net primary photosynthesis. Determination of net primary photosynthesis, NP. N is equal to volume of dissolved oxygen in light bottle determination of net primary photosynthesis, NP. N is equal to volume of dissolved oxygen in light bottle minus initial volume of dissolved oxygen. We got NP. Determination of respiration, R. Initial volume of dissolved oxygen minus amount of dissolved oxygen in dark bottle. 1.6 minus 0.4 is equal to 1.2 mg per liter. Then determination of gross primary productivity, GPP is equal to GP into 0.375, 4 into 0.375, 1.5 gram per meter cube per 3 hour. Determination of gross photosynthesis, GP is equal to NP plus R. Here GP into that thing. Gross photosynthesis, NP plus R.